All right. So, this is the introduction. So, microbiology studies small things. How small? The smallest ones are, that we're going to talk about, are viruses. And usually to see them, you need an electron microscope. Now, is that true for all viruses? No, it isn't. The largest viruses, not even the smallpox, but viruses like Pandora virus, you can fairly clearly see in the light microscope. Does that make sense? So we can say that in general, you need an electron microscope to see viruses. But there are exceptions from the rule. For other microorganisms, such as bacteria, oh, for the love of God, bacteria or archaea or protozoa, okay, usually you can use light microscope and it's fine. Some bacteria are super small, so you better be using electron, but overall that would follow the suit. So far, any questions? We're good? Moving on. So in general, this is the sequence of sizes of microorganisms that we're talking about. Viruses are smallest, helminths are the largest. As a matter of fact, helminths most of them can be observed with the naked eye. Which brings us to the conversation about main structural features that characterize each individual group of microorganisms. So first of all, we can describe microorganisms as being pro or eukaryotic, okay? You may remember that eukaryotes, they have a nucleus and the membrane-bound organelles, while prokaryotes do not. Clear? No? Okay. Now, prokaryotes, without an exception, are unicellular. There are no multicellular prokaryotic organisms. Why? Well, it's a good question. Most likely because prokaryotes cannot establish sufficient communication between the cells. Now, the branch of microbiology that studies bacteria and archaea, those prokaryotes, is called bacteriology. And then we have a bunch of eukaryotic organisms. Algae, which can be unicellular, like this, Emiliana huxleyi, or multicellular, like kelp. And the branch of microbiology that studies them is called phycology. You should know that. Protozoa, their feature is that they are eukaryotic, which are always unicellular. There's a definition. And then, of course, we have fungi. Fungi can be unicellular, like yeast, or multicellular, like mold. And the branch of microbiology that studies them is called mycology. Finally, helminths. Those are the only organisms in that list that are always multicellular. Again, the eukaryotes. Now, what about these guys? What about viruses, right? So if you look at the viruses, like this filovirus, viruses do not have cell structure. They don't have cells, which means you can't really characterize them as being unicellular, as being pro or eukaryotic. All good so far? Clear? Now, you don't need to know the examples. It's a rule. So how do we classify microorganisms? Originally, the classification of living organisms, well, originally it was Carl Linnaeus, but Whittaker classification that was widely accepted mid-20th century relied on 5 kingdoms: Monera, protists, plants, fungi, and animals, which were based on the observable characteristics, such as structure or ecology. Now, I think you don't have any problem with identifying members of fungi or animals on plants, but what about Monera and protists? So Monera are all prokaryotes, and protists are single-celled eukaryotes. Now later, I believe in late 1970s, Carl Rose developed his own system based not on structural similarities, but on the genetic similarities between very conserved, very stable genes. And he demonstrated, along with his colleagues, that all organisms, based on their genetic relationships, evolutionary relationships, can be grouped into 3 domains: bacteria, archaea, and eukarya. If you look at this phylogenetic tree, effectively 23andMe of nature, you can see that this last universal common ancestor gave rise to bacteria, and another branch revealed archaea and eukarya. Paradoxically, eukaryotic organisms are closer to archaea than both of them are to bacteria. Effectively, archaea and eukaryotes are brother and sister, while bacteria is their cousin. We doing all right so far? Now, what about naming? We use binomial nomenclature for all living things, which effectively excludes viruses. When you look at the name of the species, such as Giardia lamblia, these 2 words represent, well, again, the name of the species. Within that name, the first word, Giardia, refers to the genus. It is applied to all living organisms from bacteria to humans. The first word represents broadly the genus, and both words name the species. Viruses have the most awesome names. Plant viruses in particular, this is the real virus, which the name describes what it specifically does to tomato plants. Okay? So I kind of cut on the history of microbiology, just FYI, microbiology. The idea that diseases can be transmittable appeared in ancient times among Greeks and then Romans, and Romans were the first civilization to build a proper sewage system, which greatly reduced the diseases associated with the gastrointestinal tract. Okay? But the ultimate question that biologists had to answer is whether life can appear from non-living things, so-called spontaneous generation. The first person to question that was, seriously question that, was Italian scientist Francesco Redi, who held meat in 3 containers. One container was open, so no doubt that there were some maggots. Flies were landing and laying eggs. Another container was tightly sealed, there were no maggots, which from our perspective makes perfect sense. There's no contact. And then there was this. So there was an exchange, so to say, in terms of the gas from meat to outside world, but flies nevertheless couldn't land on meat. So even the gauze that prevented the contact of living things with non-living meat demonstrated that non-living objects probably cannot give rise to life. And then there was famous Pasteur experiment here, in which he compared two flasks with curved necks. These flasks were effectively sealed by this water seal after being boiled. So this flask, he snapped the neck and exposed the content that was sterile to the environment, and eventually, in a few days, he saw the growth. To my knowledge, this flask, which neck remained intact, is still presented in Pasteur Institute in Paris. This experiment was done 130 years ago. That's pretty impressive. Okay? So that was pretty much the final blow into spontaneous generation theory. And then a bunch of Germans came up with an idea that actually the building block of any organism is a cell, and a cell can be derived only from another cell. So here we go to the modern biological concepts. Now the connection, the actual solid connection between the idea of disease being caused by infectious agents started in the mid-19th century with Ignaz Semmelweis, Hungarian by nationality, physician who worked as a chief medical officer, let's call him this way, of a hospital in Vienna, Austria. You noticed, so back in the days, a lot of women postpartum developed sepsis. It's called puerperal fever. You can see that in some years, it was 1/3 of all women who developed sepsis. It's 19th century, there are no drugs. You can't really treat it. So those women were doomed. Semmelweis was not happy about that, and he noticed that women who were taken care of by midwives had better chances at survival. So he was wondering what's going on. And turns out there was one location in a hospital which was often visited by residents but not midwives. Anyone wants to venture a guess? Nope, not OR. More notorious.
The bathroom?
The morgue. Residents had to practice on cadavers. And they would practice, no gloves, folks. They would do the dissections and go back and deliver babies. Sounds incomprehensible by the modern standards. So Semmelweis simply introduced hand wash with chlorine, and the rate of sepsis dropped. Okay? That make sense? Later, same century, Joseph Lister, the British surgeon, introduced this interesting contraption that sprayed carbolic acid, effectively water solution of phenol, on the patient's skin and wounds during the surgery. Little detail, just FYI. It was the first antiseptic technique, or aseptic technique. Right? Make sense? There was no anesthesia back then yet. Yeah. Kind of sucked, right? Well, before it was worse. No anesthesia and no aseptic technique. So these were two pioneers in microbial control in a hospital. But at the end of the day, it was Robert Koch who proposed his famous Koch's postulates, allowing future researchers to establish the connection between a microorganism and the disease that it potentially causes. Koch himself discovered Mycobacterium tuberculosis. He discovered Vibrio cholerae, bacterium that causes cholera. Something else. I don't remember the third one. Okay? Good? So obviously, microorganisms are important. At some point, I was trying to come up with the location on Earth which does not have any microorganisms, and I actually figured it out. Anyone wants to guess? Which places on Earth do not, sterile, completely? Once again? Can't hear you. No, absolutely not. No. It's not real, but there are microorganisms. Well, you get, like, seriously, dude, yeah, nothing, yeah. I mean, Earth, like the stuff that we can get to. Nope. There are many corpses there, by the way. Yeah. Volcanoes, something hot, not survivable, right? Cold is not a problem, the heat is. So microorganisms are everywhere, except volcanoes. I'm going to say the triviality. Microbes cause diseases. Right? Right? They do. I'm not going to go through this table, although it is very interesting since our time is somewhat limited. I just want you to understand that, especially in developing countries, infectious diseases is the #1 cause of mortality. Okay? If we take Africa, what do you think is the main killer? Malaria is about third, fourth. Huh? No. HIV. #1. Tuberculosis. Go ahead.
You're going to say it, right?
What?
Like the answer to the question.
Yeah, HIV is the #1 cause.
Oh.
What question?
Oh, I thought you were asking me.
I was asking, what do you think? There were proposals that it's malaria. Malaria is top 3.
I was going to say like something with E. coli.
Nope. So, infectious diseases of gastrointestinal tract in general is in top 5.
When you say infection disease, you mean the disease that caused by microbes? Like bacteria and
Viruses, fungi, not diabetes. Diabetes is not an infectious disease. Or cardiovascular event is not an infection. Does that make sense? So, they cause a lot of deaths. Okay? You can look at this table when you have time. It's kind of interesting, the dynamics in the U.S. Like we had the mortality from vaccine-preventable diseases in 1980 was 2.1 per 100,000. 2014, it's 0.8. Ideally, it should be 0 or very close to 0, but, you know. I'm looking forward to the current Department of Health and Human Services administration because it's going to be a very, very exemplary shit show in the next 2 years. Bacteria can be useful. For example, on this graph, you can see an observation of the wine microbiome changes during fermentation, which may allow people to track the perfect wine, to mature it the best way possible. This god-awful and very interesting chemical pathway here demonstrates that bacteria can be used to produce solvents or butanol, the kind of alcohol that can be used in cars instead of gas. It's still experimental. And bacteria, of course, since they have to compete with other bacteria, can produce antibiotics, chemicals that kill the competitors. Okay? All good, folks? Everybody? All right. Which brings us to the first group of pathogenic microorganisms. I peppered the course with a bunch of pathogens to make it a little more interesting. So these are Clostridia. All Clostridia have something in common. They are obligate anaerobes. What does that mean?
They cannot have oxygen.
Correct. They cannot survive in the presence of oxygen. And they form endospores, structures that protect their genetic material, making them very hard to control chemically or physically. Okay? All good? So 4 common Clostridia species that cause human disease, presented here. So Clostridium botulinum, unremarkably looking bacteria here, causes the disease known as botulism. which is a flaccid paralysis because of, interruption in nerve transmission. People cannot contract their muscles. All good, folks? No? All right. Clostridium tetani, on the other hand, causes spastic paralysis known as tetanus or lockjaw, when all muscles are contracted. Okay? Now, for any infectious agent that you will see in the course, you need to know, what it causes, what kind of disease, how it is transmitted, and whether or not there's a vaccine. So, Clostridium tetani is the only one that can be prevented by vaccination. We're good? If you go into a nursing program in your clinical practice, your chances to see either of these two are very slim. Botulism is rare, and tetanus is rare too, thanks to vaccination, but who knows? Okay? Questions? Next goes, both of them are environmental. Clostridium botulinum, though, mostly is transmitted by eating food that is contaminated, but still environmental. Clostridium tetani and Clostridium perfringens, they come from the environment, from the soil, pretty much. Clostridium perfringens causes gas gangrene. That stuff, you'll see. Remember that these microorganisms are anaerobes. They want low oxygen environment. People with diabetes have a pretty bad perfusion, blood flow, in their limbs, especially in the legs, feet, okay? So if there's a wound and Clostridia get into the wound, it's a perfect environment. That make sense? So you're going to see gangrene in people with diabetes. And finally, C. diff. I understand a lot of people here work in some kind of healthcare settings, so you're familiar with that scourge. It causes colitis, perforation of the large intestine. And 25% of people here in this class have Clostridium difficile in their guts. It's just a fact, it's a known fact. Now, why those people don't have C. diff? Because they also have healthy microbiome that prevents Clostridia from multiplying. Once a patient like that receives an aggressive antibiotic treatment, it wipes off the good bacteria, Clostridia flourishes and produces toxins that ultimately lead to that colitis. Good? Now, treating C. diff with antibiotics sounds kind of counterproductive. It's like extinguishing fire with gas. You probably can do that, but it's a risky strategy. So now the treatment of Clostridium difficile is focused mostly on the replacement of microbiome through fecal transplantation. Now, fecal transplants now are made from freeze-dried human feces. They're all tested, to my knowledge. I don't know if now, but a few years ago, Cleveland Clinic paid $40 per dump. They tested it, obviously, but you could get some money for the poop. Before that, it was enema, so fecal suspension from the donor was delivered through enema, which is, and in the very beginning, it was just people were drinking fecal suspension, which I know sounds disgusting, but it also defeats the purpose. Gut microorganisms usually do not survive your stomach, which is acidic. That's why they changed to enema, because it was a more direct way to populate. Now it just pills. Okay? Sounds good? So Clostridium difficile is also environmental bacteria. You can find it in the soil, but it mostly resides in the guts of humans and other animals.
So treatment for C. diff is
Fecal transplant for the most part, yeah. Now we're going to jump back to a quick review. So here, water and pH. Things to know, things to remember. The idea of a hydrogen bond, the bond that is formed between partially negative oxygen in one water molecule and partially positive hydrogen in another. Okay, just know what a hydrogen bond is. They're very important in formation of DNA proteins. And understand the whole pH scale. Do not memorize this. 7 is neutral, above 7 is basic, below 7 is acidic. Do you follow? If you increase acidity, pH falls. If you increase alkalinity, pH rises. Good? Any questions? All right. Carbs. Things to understand. The composition. This is not a chemistry class. I am not asking you to remember any of these formulas. Okay? I will not ask you formulas. I will not ask you the composition of maltose or sucrose or lactose. I want you to understand at the basic level that we have a bunch of monosaccharides which you can combine together in a chain called polysaccharide. Another product of this reaction is water. This is why it's called dehydration synthesis. And if you want to break this chain back, you need just to add water and some enzymes. Okay? We good so far? So functionally, mostly energy and structural roles, it's just cell wall. Obviously, you can find carbohydrates and other molecules, but I'm not going to focus too much on that. Lipids, 3 types I want you to remember. First are triglycerides. Why do we call them triglycerides? Triglycerides consist of glycerol residue and 3 fatty acid residues. All lipids are nonpolar/hydrophobic. The use for triglycerides is mostly energy. So you need to know the composition, glycerol and fatty acids. When we get to phospholipids, we add one more component into the mix. Instead, one of the fatty acids, you have a residue of phosphate. So it's phosphate, glycerol, and 2 fatty acid tails. Turns out that this part of the molecule, the part that consists of glycerol and phosphate, is hydrophilic or polar, while the tails that consist of fatty acids are hydrophobic, i.e. nonpolar. Any questions? What is the function of these guys? The function of these guys is to form a membrane, as we will see very, very soon. Okay? And finally, this interestingly looking polycyclic lipids, sterols on the left and hopanoids on the right. Their function is to reduce the viscosity of cell membrane. Cell membrane is not a solid structure, it's not a wall. It's basically very viscous liquid. If you add sterols or hopanoids, it becomes less rigid, more fluid. All right? So what's the difference between the two? Well, sterols, they regulate the membranes of eukaryotes. Hopanoids do the same stuff in bacteria. Any questions before we move to proteins? Sterols, they regulate the fluidity of the membrane in the eukaryotes, while hopanoids do the same function but in bacteria. Now let's get to the proteins. Functionally, proteins do stuff. 99% of cellular function is carried out by proteins. So the basic building block for proteins is an amino acid. If you look at amino acids on the right-hand side, you will see that they all have the same skeleton, the same part, and then they differ by their side chains, highlighted here in blue. So this difference in side chains has a very important consequence from the functional standpoint. If you take two amino acids and join them, they will be joined by the peptide bond in the reaction of dehydration. Very good so far. You can keep adding amino acids, forming tri, tetra, pentapeptides, and eventually you will form a long chain of different amino acids. Make sense? This is called a polypeptide and represents the primary structure of the protein. Because amino acids are different and their parts interact with each other through hydrogen bonds, for example, the polypeptide chain will start folding. And it will fold into a secondary structure, two main elements of which are alpha helices, where amino acids, the polypeptide is folded, well, as a helix, or beta sheets, where polypeptide is effectively folded like a sheet, like that. Now, the interactions between the side chains of amino acids will determine the tertiary structure of the protein, the globule. So far making sense? For many proteins, that's going to be it. Many proteins represent just one polypeptide. They're usually relatively small. But a lot of proteins, especially enzymes in the cell, have multiple polypeptide non-chemically merged together. They do not form specifically chemical bonds, they interact because of the charges and so on and so forth. Okay? This assembly of several polypeptides is called quaternary structure of a protein. You must know what represents each structure and what are the main elements like alpha helix and beta sheet. So far, good? We effectively established that protein structure is defined by the side chains of amino acids. Do you agree with me? Interaction between side chains will define that. You know, you have two polar side chains, they may interact, two non-polar, they may interact, just like that. So if you change an amino acid and change the side chain, you can potentially alter the protein structure. And if you alter the structure, what else do you alter? The F word. You alter the function. The whole concept of mutations and subsequent amino acid substitutions, that's, there you go, that's, changes in a polypeptide. Another thing, a lot of those interactions that we observe within proteins depend on the chemical composition or the temperature. It means that the structure can be disrupted. Protein structure can be disrupted. We call it protein denaturation. Two glaring examples, the egg drop soup, Chinese cuisine, which is very acidic, and when the egg is going there, it denatures, the proteins denature. And second example is when people try to make the hair from curly to straight and vice versa. It's done with temperature and chemicals. Does that make sense? And it's not permanent because the proteins in the hair will eventually assemble its initial shape. Okay? Finally, nucleic acids. So these guys consist of nucleotides, 4 types. A, G, T, and C for DNA. For RNA, T will be replaced by U. Please do not focus on that now. It's Unit 2. For our intents and purposes, DNA is double-stranded, RNA is single-stranded. The fundamental feature here is that the strands in DNA, for instance, they are complementary, which means if you have A in one strand, there's going to be T across it in another. If you have G in one strand, there's going to be C across it. Okay? So that allows coding. That allows the concept of a message, and what's important, this message can be copied and passed down from generation to generation. And this brings us to 2 most important concepts in microbiology: gene expression and evolution. So, when we are talking about the genome of an organism, we talk about the information about its traits, right? So like bacteria can have a gene for antibiotic resistance. This is a phenotypical trait. Bacteria can have a set of genes that will result in the production of capsule. Capsule is a phenotype. Am I making sense? So phenotype is defined by genotype. But I want to be a little bit more specific. So let's say we talk about a bacteria that everybody knows, E. coli. It has 5,000 genes. A gene encoding particular trait. We're good? Do you think that all those genes are expressed, all those genes generating proteins at the same time? Probably not. So, gene expression responds to changes in the environment, what authors attempted to represent by this flowchart. But I think I came up with a much better analogy. It's an analogy of cooking. So imagine that over generations in your family, you accumulated 5,000 recipes. They stored in a book. This book is in your living room, is your cherished possession. It's effectively invaluable because it's your family heritage, right? That's your DNA. Okay? All the recipes. And tomorrow on Friday night, your friends are going to visit you, and your friends is a pretty interesting and varying bunch. Okay? You have a person with celiac disease, so gluten is out of the question, right? You have a person with lactose intolerance, no lactose. You have a Muslim person, so everything should be halal, right? You should exclude certain meats. You have a guy who's Hindu, so other dietary restrictions. And then you have a vegetarian friend. So you are facing certain environmental challenges. Are you going to cook all 5,000 dishes? No. You're going to choose certain, what fits best. Okay? And in response to this challenge, the changing environment, you're going to start making a meal. Do you follow my train of thought? So you decided on the recipes. Now you need to bring those recipes in the kitchen. Remember, 5,000 recipes. It's a pretty massive cookbook. And it's super valuable. Would you take it to the kitchen? Like the book itself? What would you do then, if not?
Take a copy.
Yeah, but how do you transfer them?
You just write it down.
Write it down. Well, come on. It's old people's talk. You're going to take a picture with your smartphone, right? And take a picture, take it to the kitchen. The taking a picture is transcription. Okay? Well, I'm going to put picture here. That's a picture of the recipe. So you effectively have the same, you have text in the book, you have text in your smartphone, it's both texts. Nothing really changed. Then you start following the instructions, and you are cooking, making a meal. That's protein. You convert the instructions of the recipe into the product of an actual edible meal. Your friends aren't going to appreciate if they will come in and say, well, I have pictures on my phone, look, we can eat them. No, you can't. So that's the idea. You have to get all the way to proteins, for the most part. Does that make sense? Why I like this analogy? Well, first of all, because it's mine. Second, because it works really, really well. If you start thinking about the fate of RNA in the cell, those images on your smartphone, those recipes, are you going to keep them forever? Same's true for RNA. RNA degrades pretty quickly. Or let's say you copying by hand and you make a mistake. Okay? Is it a catastrophe? I mean, not really. You're going to screw up one meal, you're going to cook it again. If you put a mistake, if you put change in the cookbook, that's a different story. Okay, you can give a copy of the cookbook to your kids. It's passing DNA down the generation. Does that make sense? Awesome. Second concept is evolution. There's Dobrzański, who was a Polish biologist, evolutionary biologist, immigrated to the United States, I think in the 1930s when Germans started working in Europe. He said nothing in nature makes sense other than the light of evolution. So if you start asking yourself, why does that happen? Why is that microorganism, why does it look like that? The universal answer is evolution. The only problem, we don't know the history. Does that make sense? But I'm not even going to focus on antibiotic resistance when we talk about evolution. I'm going to focus on something else, the pandemic that we had recently. At the beginning of the pandemic, several pharmaceutical companies released antibodies, drugs that effectively reduced the ability of coronavirus to infect humans and reduced mortality, especially in elderly patients. Okay? So guess what happened in about 6-8 months? It's selected for the individual viruses that could avoid those antibodies. It was inevitable. Everybody understood that it's not going to work forever. People are buying time. But that shows you, like, since then, those original antibodies, if you will try to use them now, they have no clinical effect. It's been demonstrated. Virus evolution is random, but it's driven by selective pressure. So when you look at something that happens in cells, you have to ask yourself, what kind of selective pressure are we dealing with now? Well, good. So now we're going to get into the, grind of it. Morphology. I'm not going to read this table to you, okay? I can tell you what kind of question you can expect. What is the term for a curved rod? Vibrio. You will effectively have to match the term and the description. Does that make sense to everybody? So I'm not going to spend time on this. Obviously, some genera of bacteria named just because of the shape, like Vibrio bacillus. Now, how do cells maintain their cell structure? Well, here we're talking about osmosis. So first of all, what is osmosis? Osmosis is a diffusion of water towards the higher solute concentrations. Notice that we're not talking about the concentration of any specific solute. We're talking about the total concentration of everything, which is called osmolality, which depends on the number of solute particles, but not the exact chemical nature. Now, if osmolarity inside the cell and outside the cell, if it's equal, we call the environment around the cell isotonic. You probably remember that term from previous biology classes or chemistry classes. If the environment has a greater concentration of solutes than the cell, we call such environment hypertonic, and what happens is water leaves the cell, forcing it to shrink and often undergo osmolysis. Finally, if the cell has more solutes inside than the solution outside, we call such environment hypotonic, and in this environment water instead moves into the cell, resulting in the cell swelling. In case of many bacterial and fungal cells, it's not such a huge problem because they have cell walls which prevent bacteria or fungi from exploding. But what about protozoa that do not have, don't have it here, but I'll just talk about it. What about protozoa that do not have a cell wall? They usually live in hypotonic environments. So now I want you to, for a second, imagine that you are a tiny protozoan cell, just one. No cell wall, just a membrane. Can you imagine that? And the water constantly comes into your cytoplasm. It flows into the cell. You're supposed to explode, and yet you don't. What do you do to not explode? You can't. You have nothing to absorb. Say again? Yeah. It's like a sump pump in the basement. Does that make sense? Protozoa constantly pump water out. Water that comes in. This is why if you look at protozoan cells, they're constantly moving. They're always eating. They're always in the hunt because they need so much energy. Good? Another question that I want to ask you is this. You're probably familiar with a body of water, or rather brine, called the Dead Sea in Israel. It's extremely salty. And yet, there are bacteria and archaea that live in these salty waters. How do you think they manage to survive? Let me guide you through. Go ahead. Yeah, but how? Of course, they have adopted, but how?
They somehow ignore the osmolarity.
You can't ignore the osmolarity. It's like ignoring gravity. You can do it, but just once. Okay? Huh? I mean, that is the answer that covers everything.
They just transaction.
Say again?
They just do some transaction between the molecules and make some.
When we talked about hypertonic and hypotonic, did I ever mention a single number? No, I didn't. Because it's a relative parameter, right? Hypertonic environment simply means that there's more solids outside than inside, but no numbers. Now, cells would feel totally comfortable in isotonic environments. Would you agree? So those cells that live in Dead Sea effectively have the same concentration of solutes inside that's outside. They don't have to be the same solutes, though, but they maintain the same osmolality within the cell. If you will try to put those cells in like lake, they'll explode immediately. Even cell wall is not going to save them. Now, we're going to talk about the structures found in prokaryotic cells. Every prokaryotic cell has these four structures. I want to urge you from when we use the term, it is the exact term. Like nucleoid isn't nucleus, although they sound pretty similar. So all prokaryotic cells have nucleoid. Remember, there's no nucleus. A nucleoid is just the area where DNA is. Okay? They have cytoplasm. That fills up the cell. They have a membrane that surrounds the cell, bounds it. And they have ribosomes, which are small non-membrane bound organelles consisting of RNA and proteins, which translate mRNA, synthesizing proteins. Okay, everything else